nervous system versus the endocrine system. But otherwise, they're very similar. They're both messengers, and they both have to bind to a target to have an effect. Okay, without a target, both neurotransmitter and a hormone are useless. Okay, so they're kind of similar in that sense. And let's see, slide 5. We talked about two major groups based on classification of action, whether they're polar hormone or non-polar hormone, right? We said polar hormone is the same thing as water soluble, right? So if you're water soluble, you're not able to pass through the cell membrane because the cell membrane mainly has lipids, right? So you're not allowed, this polar hormone is not allowed to pass through. So instead it has to stay out, and its receptor is also going to be on the surface. It doesn't make sense for its receptor to be inside if that hormone can't get inside, right? Okay. And the opposite, non-polar hormones are lipids basically. They're lipid soluble or lipophilic, however you want to call them, and they're not soluble in water. So they have no problem getting through it. They'll go straight through the cell membrane, and therefore if they can get straight through the cell membrane, then their receptor must be where? In the inside of the cell. Okay, because again, it doesn't make sense. They can get inside, but the receptor is hanging out on the surface. Okay, so we can group them that way. Okay, and then let's see where we left off. You guys are a little bit behind from my other classwork. So we were talking about multiple effects, right? I think when we met last time. How you can have several hormones targeting the same cell, and they'll have obviously a multiple effect on that target. And we're doing like 2 hormones at a time. We talked about synergistic effect between 2 hormones where combined, when they tag team, they have a really much bigger combined effect than as opposed to just adding them together. It doesn't work that way. They enhance each other basically. Like I told you this example, if epinephrine increases heart rate by 10% and norepinephrine by 20, don't think that together, just add these 30%. No, it's just much bigger, like 70, 80%. Okay, their combined effect is much greater than if we were to sum them up. Okay, that's synergistic effect. In order to have a synergistic effect, both the hormones have to have the same function, and they do, which is increasing heart rate. Okay, but other hormones, like if they don't have the same effect, it's not a synergistic effect. Okay, it can't be. And I told you about antagonistic effect, that they're opposites of each other. They kind of inhibit each other. So insulin and glucagon are an example, right? Insulin will lower the blood glucose level, and glucagon will raise it, right? Remember I told you when glucose is gone, you make what? Glucagon. So when blood glucose levels are low, technically kind of gone, then glucagon will be secreted to do what? Raise. Okay, so they're opposite of each other. And here's another pair that's kind of opposite, calcitonin and PTH. Although you don't really have to know PTH, I didn't talk about it. But calcitonin, I believe I did talk about it. What what gland releases calcitonin?
Thyroid gland.
Which one?
Thyroid.
Thyroid gland, right? Because people always think, only thyroid hormone is released from the thyroid gland. Not true. Also calcitonin. That was from the last time we met. Okay. Alrighty. So that's the antagonistic effect. All right, so I think we're here with permissive effect next. So let's start here. The button's on, I can't reach that. I don't know if you had a stick, I could like try to, a little thing there I can turn on and off, but I can't reach that.
These shirts don't have wheels on them.
Huh?
These shirts don't have wheels on them.
You want me to step on one, or if I can reach
No,
it?
I'm sure this is.
Yeah, okay, sorry. I don't see a huge mistake. Okay. Yeah, I'm sorry, I'm not, you always said it'll turn on. I'll have to remember to tell them to put a work order for that projector. Okay. Alrighty, so we're talking now about permissive effect, okay? And with permissive effect, I think the name is kind of useful when we think of permissive effect. It's like one hormone is permitting the other one to work, right? Or it kind of allows, or one hormone enhances the effect of the other one. So by itself, that first hormone really doesn't do much until that second hormone kind of is present and it kind of permits it or allows it to work, right? So for example, here we have thyroid hormone and epinephrine, okay? And what's gonna happen here is that thyroid enhances or permits the effect, T3, thyroid hormone, enhances or permits the effect of epinephrine. I'll show you what I mean by that in a moment. So it enhances or permits the effect of epinephrine. So when we talk about, well, if we consider lipolysis, this process called lipolysis, which is basically breaking down lipids, epinephrine can sort of kind of do it, but by itself, if you just have only epinephrine present, it does it weakly. It doesn't really do a good job of it, of breaking it down. However, if in the presence of thyroid hormone, that same epinephrine that used to by itself only weakly break down lipids, it will strongly now be able to break it down. Okay, so again, that's why I told you the thyroid hormone T3 permits the effect of epinephrine, enhances or permits it. Okay. What is thyroid hormone doing though to permit? How does this happen to permit the effect of epinephrine? And it all has to do with receptors. So what happens here is that thyroid hormone actually increases the number of epinephrine receptors on that target cell. That's what thyroid hormone does. So it kind of does epinephrine a favor by having more receptors. And again, you know hormones have to bind receptors. And now epinephrine, that's why it has a much greater effect. Because again, thyroid hormone allows for more receptors for epinephrine. Okay? Let's talk about some other effects. We're going to talk about priming effect and then desensitization. So let's talk about priming effect first. The priming effect is also called upregulation. And what we mean by upregulation, this simply just means increasing the number of receptors on a certain target cell. Okay, for a certain hormone, it could be the same hormone that we're dealing with or another hormone, but again, it's basically increasing the number of receptors. Because the more receptors you have, the more hormones can bind. And the more hormones that bind, the greater the effect is going to be on that target cell. Okay? So, for example, let's say I have my cell right here, and we have one receptor for epinephrine. Here's epinephrine. We have some epinephrine here. Okay, this is all epinephrine, and this blue receptor is the receptor for epinephrine, right? Okay. And then we have some other receptors on it, on the same cell as well, not for epinephrine, for something else. More receptors. Okay, and then in my next picture over, time lapse, here, let's go back to blue, because color does kind of matter. Right? So this time, we have more receptors for epinephrine. Before we had one, now we have 3 receptors for epinephrine. And then let me put back my red ones here, and then I'll explain everything to you. And now these red ones now are thyroid hormone. These are all thyroid hormone binding to their receptor. So this is T3, for example, right? So in other words, what's happening here, there's no little red circle, so there's no T3 present, right? So in other words, we have a cell, and it has receptors, 3 receptors for T3, but there's no T3 present here, right? And we only have 1 receptor for epinephrine, and there's only epinephrine here. So in this case here, the first picture, this is only epinephrine present, no thyroid hormone, correct? Second picture, you have both, what color is this? One color. Second picture, you have both epinephrine and thyroid hormone. Right now, thyroid hormone is present, and what did thyroid hormone do? It increased the number of receptors. Right here, when we had one receptor, here we have 3 receptors for epinephrine. So the presence, yes, the presence of T3 increased the number of receptors for thyroid hormone. I'm sorry, I keep saying thyroid hormone, epinephrine. Thyroid hormone increased the number of receptors for epinephrine, and now epinephrine can have what? A greater effect. Does it make sense why it has a greater effect? Because you have more hormone receptor interactions here on this cell, as opposed to the first one. Here, only one epinephrine combined, because you only have one receptor. Here, all of them combined, because you have more receptors. Why do you have more receptors? Who's present now? Thyroid hormone is present. It increased the number of receptors. That's priming effect, upregulation. Sometimes priming effect can also be, may also be an example of the permissive effect if the two hormones are involved. So this is priming effect. Do you agree with me? The increase the number of receptors. Is this also permissive effect between epinephrine and thyroid hormone? Yes, it is, because now I told you epinephrine is going to have a greater effect because you have more receptors for it, right? And I think that was even the example for if you go back to permissive effect, right? It is thyroid hormone and epinephrine, so you kind of knew it, but I want you more to understand, why that is, right? You have a greater response because, again, T3 and epinephrine, in other words, T3 increases the number of receptors for epinephrine. And it's going to lead, like I told you, to increase response. So this is both a priming and permissive effect. This is priming and permissive. Okay? Because again, we covered permissive over here. Okay, any questions on priming effect? Okay, let's go to desensitization effect. This is called not upregulation, but downregulation instead, right? And this time it's the opposite. You decrease the number of receptors. So it's kind of like, I could probably use my same picture and then now just go this way, right? Make it kind of go in the opposite way. I went from 3 receptors to like 1 receptor. So they're like opposite of each other, right? And again, if I have less receptors, again, let's just say for epinephrine, then the response is also going to be decreased, right? And this usually happens after prolonged exposure, right, to that hormone. You sometimes, if you don't want that effect anymore, the best thing to do is to get rid of the receptor. If there's no receptor, the hormone doesn't have anything to bind to, and that hormone cannot cause an effect at all. Because again, a hormone will cause an effect on the target cell only if it binds a receptor. Get rid of its receptor, it's just hanging out in the blood, not going anywhere, not doing anything. Okay, so yeah, this is what happens after prolonged exposure. And again, this also is useful to prevent overstimulation of that target cell. Get rid of some of the receptors and it won't be stimulated as much. Because a cell that has a lot of receptors is going to be more stimulated than one that has less receptors on it. Okay? Alright. This, if you printed your notes like earlier when I first put them out, maybe I only had thermoregulation, but I added this part. I don't know if your notes have that. This is negative feedback regulation of endocrine pathways. I added that because we're talking about 2 negative feedback pathways that you see here from this pathway. Okay, so I just added that, I think, last night or this morning. I remember that. I was going through this all over again. Okay, so thermoregulation, we're going to talk about. And what that is basically the ability of an organism, a human, to maintain a constant internal body temperature. And by constant, this is kind of dealing with homeostasis. It's like within a normal range. And we talked a little bit in lab, but from an endocrine perspective, this is how it works. Several tissues are involved and several hormones are involved. So if you start here at the top with the hypothalamus will release this hormone called TRH, which stands for thyroid releasing hormone. The R stands for releasing. So thyroid releasing hormone. Okay, TRH. And then TRH is going to stimulate the pituitary, and the pituitary will secrete TSH. Which pituitary releases TSH? Anterior. Remember it was part of the flat pig? This is the T in flat. So this is the anterior pituitary, right? TSH, like the name tells you, thyroid stimulating hormone, thinking of homework, right? Thyroid stimulating hormone, the name tells you it's going to stimulate the thyroid, and it will, and thyroid now is going to release mainly T4 and T3. Okay. For thermoregulation, calcitonin is not involved. What is calcitonin involved with, by the way? Lowering what?
Calcium in the blood.
What are the first three letters of calcitonin?
Calcium.
So calcium, it's involved in decreasing calcium. We're not talking about calcium here, we're talking about temperature. And calcitonin doesn't play a role in that. T4 and T3 do. Okay, I'm just saying all three of those came from the thyroid gland though, right? But the thyroid hormones are the ones that are involved in temperature. So anyhow, you release now T4, T3. And T4, T3 is gonna, its target is gonna be these cells or tissues. Okay, they call them thyroid hormone sensitive cells, whatever, tissues. And what these tissues are gonna do, because I kind of ran out of room, what these tissues tend to do, they increase metabolism. Okay, the metabolism in these tissues or cells is going to increase. And you know already, when you increase metabolism, what does this lead to? Think of cellular respiration. What does cellular respiration make at the end? Energy, right? In the form of either ATP or what else? Heat. And we're gonna focus a little bit more on heat. So it's basically all of this system that we've talked about regulates body temperature or heat. That's what thermal regulation is referring to. Okay? And that's one of the functions, of thyroid hormone is increasing metabolism, and you're gonna end up making a lot of ATP and heat. All right? Let's say you're hot enough, you don't wanna like burn to death, right? Because you have to be in a certain range, right? Now you're at the peak of that height, right? Anything really above that, now you're talking about possibly fever now. So your body's not gonna allow that. So what you're gonna have to do is negative feedback on some of these tissues. So if you wanna stop the heat, then stop making thyroid hormone. Because if you have thyroid hormone, what is gonna happen? All of this. And you're gonna keep making heat. You're gonna burn to death, you're gonna fry your brain, so you need to stop. So how do we stop thyroid hormone from being made T4 and T3? Thyroid hormone will come back and communicate with two areas. It comes back at the hypothalamus and tells the hypothalamus, hey, stop making TRH. Why does it tell it to stop making TRH? What happens if you have TRH? All of this. If you have TRH, you're gonna get TSH, you're gonna get thyroid hormone, and you're gonna, again, increase metabolism in the tissue. Does that make sense? You need to stop TRH. Another place you can stop is the TSH. So the thyroid hormone tells the anterior pituitary, hey, stop making TSH. Because if we don't stop making TSH, if we keep making it, it's gonna keep stimulating the thyroid gland to make what? Thyroid hormone. And you see how this whole system is gonna increase the heat. So you kind of have to put the brakes on it. That's negative feedback at these two levels, at the hypothalamus and at the anterior pituitary. That's how you regulate. And you stay within that normal that range. Okay, any questions on thermal regulation? No? Okay. This is basically everything that I said right there for you. Okay? Alright. The rest of this PowerPoint, we're gonna focus on water-soluble hormones and liquid-soluble hormones. Let's talk about water-soluble because they're a little bit easier. Okay? Right, and my water-soluble hormones are my polar hormones. okay, also known as hydrophilic hormones. And again, because they are polar, they cannot cross the cell membrane. They're not allowed to, right? So you already know then that the receptor has to be on the surface, on the cell surface. Okay. Let me close this door. You guys are all freezing, right? You cold? Everyone's freezing. I wore long sleeves today. It's my first time this semester wearing it. Okay. Yeah, the receptor has to be on the surface, right? So how do they work? How do water-soluble hormones work? That hormone is going to bind eventually, has to bind to the hormone receptor on the surface, and then when that happens, the receptor is activated. Just like neurotransmitters did, right? When they bound to a neurotransmitter receptor, then the receptor got activated. So now hormone receptor is activated, and when it's activated, you have this whole series of intracellular events inside, right? We call that signal transduction cascade, occurs inside, all right? And again, you have to understand hormones are chemical messengers. They have a message to deliver to the cell, and the cell's gonna need to do something, right? Whether it tells the cell, hey, start absorbing glucose, because maybe this is insulin that has a message, whatever. And the message is, hey, start absorbing glucose, all right? But the problem is that the message is outside the cell, because remember, this hormone cannot get through, right? So you rely on a series of events inside the cell to continue delivering that message. It's like this one needs two messengers. One that stays outside, and then the other one that's inside, right? It's like, you know, if someone wants to deliver a message to you, and the person out there has the message, but they're not allowed to get through this door, they can only stop at the door and then give it to me, and then I deliver it to you. So it's like a two messenger type of a system. If it's a polar hormone, we cannot get inside the cell. Okay, and there's two pathways that are used that water-soluble hormones typically use. This one you're familiar with already because we've talked about this, and this is pretty much the same one when we talked about the metabotropic neurotransmitter receptors. So this is again similar to the metabotropic neurotransmitter receptor, or receptor. They're very similar. Because in this pathway, it's also going to involve a G protein that's bound to the hormone receptor, just like last time it was the G protein bound to the neurotransmitter receptor. Okay, and the hormones that activate this pathway are LH, glucagon, and epinephrine. Okay, those are the ones that are polar. These are all polar, obviously hormones. They have a message, but the message, they cannot get through it. They have to deliver it to somebody else to take over. And this is a new one that you haven't heard of. This is a new one, phospholipase C calcium pathway, right? And the hormones that activate this pathway are TSH, ADH, and also angiotensin. These hormones here, these are involved in the kidney. This is like renal physiology. We'll come back in exam 3 to cover ADH and angiotensin, but they are hormones and they work by this phospholipase C calcium pathway. So we'll go through this one in detail some more as well, but it's very analogous to this pathway up here, the first one. All right, so again, for the polar hormones, what you have to understand is that the hormone has a message, and because the hormone cannot get inside the cell, it's going to require a second messenger pathway. Both of these are second messenger pathways. Can I put PW, remember pathways? Second messenger pathways. Again, because the hormone cannot get through, right? They at least deliver it, the hormone gets delivered to the receptor, and then that's part one, and then part two is everything that takes place now inside the cell, right? So you're familiar with this one. This was kind of very similar, pretty much the same thing when we were talking about neurotransmitters. And in that one, we were saying that it was norepinephrine. Here, you know, we're saying that the hormone, you know, it's from the previous slide, right? This can be epinephrine, glucagon, and LH, luteinizing hormone. Okay? So at first, this receptor, this hormone receptor, is not activated because there's nothing on there. G protein is not activated either. But now the hormone comes and binds here. Any one of these three comes and binds here. The receptor is activated, and also G protein becomes activated, right? And remember from last time, what happens when G protein gets activated? It breaks up. And the alpha subunit travels in the cell membrane and binds to my enzyme adenylyl cyclase and activates it. Because again, remember, enzymes exist in either inactive and they get converted to the active form, right? So now this this enzyme in the membrane is activated thanks to alpha subunit. This catalyzes a reaction that converts ATP into CAMP, cyclic AMP, right? Nothing new. And even this part really isn't that new. We know that from last time, CAMP activates protein kinase. What's new in this picture, what they're telling you is how, they're showing you now how CAMP really works, what it really does. What CAMP does is gets rid of this regulatory subunit that's attached to the inactive protein kinase, takes it away, and now protein kinase is active. That's how it does it. It just basically CAMP removes the regulatory subunit from it. And we have an active protein kinase. What do kinases do? Phosphorylate things. So, it does many things. So now, this enzyme, it's gonna phosphorylate other things, and depending on what that thing is that it's phosphorylating, it can be an active enzyme or inactive. And I'll show you on the board in the back what I mean by that. Okay, that's basically what this is saying. So this is nothing new. This is a review from the neurotransmitter part, right? This one is new, it's different, but very analogous. Instead of adenylyl cyclase in the phospholipid bilayer, now it's phospholipase C in the phospholipid bilayer, okay? And our hormones are different this time. These hormones happen to be more like TSH, like I told you in the previous slide. This is more like TSH, ADH, and angiotensin. Okay, so again, it's the same story. Hormone comes and binds here to the receptor is activated, G protein is also activated. What I don't like, they should have put, and let's put this in here, it's the same thing, alpha subunit dissociates, and let's put alpha subunit binding now to this enzyme. It's not adenylyl cyclase, this is a different pathway. Now it's phospholipase C. That's why they named the pathway phospholipase C, right? Because the pathway is named after the enzyme, right? Phospholipase C is embedded here. The other one, it was adenylyl cyclase embedded here. That's why they called it adenylyl cyclase pathway. So alpha subunit binds here, activates phospholipase C. What does phospholipase C do now? Phospholipase C will take some of the phospholipids and give you these two products here in yellow, DAG and IP3. We're gonna focus more on IP3. IP3 now is gonna bind to a receptor on this green thing is supposed to represent the ER. ER, remember we talked about the ER? Or it can also be the SR, that's in muscle, sarcoplasmic reticulum, instead of endoplasmic. We'll study the SR when we do the muscle, but this is the SR. It's analogous to the ER, same thing, okay? Anyhow, IP3 binds to here, which would be like the IP3 receptor. And then, there's IP3 receptors all over, and it lets calcium out, okay? So let's talk a little bit more about now what is this calcium doing, right? Because this calcium is out here in the cytoplasm, and what is it going to do? Where is it going to go? So for that, we have to now go to the board in the back. So I brought my tennis shoes today, right? So I'm going to do a lot of walking. I did this morning. I'm going to do a lot of walking. I did this morning. So this is the same thing that I talked to you about, right? How we activated phospholipase C because the alpha subunit bound to it, right? We had an alpha subunit kind of binding to it. That's alpha, it's bound to alpha. right? And it takes the phospholipids, because it's in the phospholipid bilayer, right? It takes the phospholipids and it's catalyzing this reaction and giving you IP3 and DAG. We don't care about DAG so much, right? So I told you IP3 binds to the IP3 receptor on the ER or SR, and the IP3 receptor is basically, it's a calcium channel, that's all it is. It's a receptor in a channel. Okay, what IP3 is, right? And then calcium goes out of the ER, right? Diffuses out from the ER or SR, right? And now the calcium is in the cytoplasm. So that's basically what I just told you and showed you up there. What is this calcium gonna do? This is a continuation. Calcium in the cytoplasm is gonna bind to this element, this thing called calmodulin. Calmodulin is pretty inactive substance. However, when calmodulin binds to calcium, it becomes active, right? So it's like a magnet for calcium. It looks for calcium in order to get activated, because by itself, without calcium, it's inactive. Very easy to remember what binds to calmodulin. How are you gonna remember it binds to calcium? Cal, calcium, so yes, it binds to calcium. And when you bind calcium to calmodulin, this complex is active. What does calcium calmodulin complex do? It activates protein kinase to give you an active protein kinase. Okay? And then this active protein kinase will hold off for a moment because it continues on and does all this other stuff. We're not there yet. It has to do with epinephrine now. Okay, but this is like a whole series of events that are taking place inside the cell. And it's all about turning on enzymes. Again, you have to understand the enzyme is originally inactive, it's off. You have to turn it on, you have to activate it, right? Just like this is activating that, and you're gonna see here, you have a whole bunch of things that activate one thing or another. Okay, so that's the story with phospholipase. See, it is one calcium. It's just not gonna sit there. There's a reason why it got secreted, because it's gonna bind to calmodulin. Okay? What I do want you to do in this slide here, let's take out this last bullet point. This is incorrect. I don't know what I was thinking. I think because I told you IP3 is a receptor and a channel, and in my head I'm thinking, oh, that's an inotropic receptor. Inotropy, first of all, has to do only with neurotransmitters. This is the wrong system. Okay? I'm talking about hormones. So this is not correct. So get rid of that one, please. That last bullet point. That's the nervous system. So this is not an inotropy. You can't understand. The inotropy has to do more with neurotransmitters. We're talking about hormones, so this is an incorrect statement. Nice try, but that was wrong. This morning I looked at it and it doesn't make any sense now. I didn't catch my error when I first did this. Okay, let's talk now specifically more about epinephrine this time. And epinephrine is involved in this process called glycogenolysis, right? What does glycogenolysis mean? Lysis means what? Break down. Yeah, I wonder what we're gonna break down. Glycogen. It's right there. The name tells you, glycogenolysis. Epinephrine can do that when it acts as a hormone. And it does this by like a two-system pathway on the same cell, but it has two different receptors that you're gonna see, right? And sometimes that's possible. You can have a hormone that actually binds to two different receptors and have the same outcome, right? But because they're two different receptors, you're also gonna have two different pathways, all in a single cell. At some point, those two pathways, though, they have a tendency to converge into a common pathway that you're gonna see up there, right? So, and that's how epinephrine works. It starts off as two separate pathways binding to two separate receptors, and then somewhere in there, there's a common pathway, and it accomplishes, like we said, glycogenolysis, and you already know what that is, right? And this occurs, the cell that it occurs on is the liver cell. Why is it the liver cell? What does the liver cell store? A lot of glucose, and that's what glycogen is, right? It's like a lot of glucose. We studied this one. It's a polysaccharide glycogen, right? Many glucoses gives you glycogen, and when you need glucose, you break down glycogen, and epinephrine can do that, right? Yeah. I just thought of something, like if you want to increase your blood glucose levels, you can use epinephrine, right? What other hormone increases blood glucose levels? Glucagon? When glucose is gone, you make glucagon, and it will increase it, right? That's a good test question too, like which two hormones increase blood glucose level, so it'd be epinephrine and glucagon. By two different pathways, two different systems there. Okay, they have two different mechanisms. All right, so again, in the liver cell, because that's where you store glucagon, that's where you store glycogen in the liver cells and also the skeletal muscle, but we're focusing on the liver cell. All right, so the liver cell actually has two types of receptors for epinephrine. It has a beta adrenergic receptor and alpha adrenergic receptor. If you printed this before this morning, I made a mistake, so make sure this is correct how I have it on the screen. I think yours, if you printed it, it had alpha receptor here and beta. Switch them. This is the correct one. Okay? If you have it now on Canvas, I corrected Canvas already. Okay, but the beta adrenergic receptor, this activates adenylyl cyclase, and the alpha adrenergic receptor activates phospholipase C. Okay. Now, before I continue, what was the other name? Remember, I told you before. What's the alternate name for epinephrine? It begins with the A. Adrenaline. The same thing as epinephrine. Remember, I think I told you that before. Adrenaline is same thing as epinephrine, and noradrenaline was norepinephrine. They're the same thing. Okay. I'm just telling you this because since epinephrine is the same thing as adrenaline, doesn't it make sense here? Beta adrenergic receptor. If I were to ask you what hormone binds to beta adrenergic, well, adrenergic comes from the word adrenaline. So yes, it's epinephrine that binds to it. So it makes sense why they named them adrenergic receptors, because epinephrine is adrenaline. Is adrenaline. Okay? But nevertheless, you have two different receptors, and again, two different pathways. Okay? And again, make sure what you're looking at now is the correct version. It's already on Canvas corrected for you. Okay? So here's my picture of my liver cell. This is a liver cell, right? And there's glycogen, as you can see, in my liver cell that needs to get broken down into free glucose. And this free glucose ends up in blood glucose now. This free glucose is in the liver cell. Now we end up, it needs to come out and end up as blood glucose, right? To raise blood glucose level. That's what you do when you break down glycogen. And who leads to all of this breaking down of glycogen so we can have enough glucose in the blood? Epinephrine does that for us by two different pathways, meaning two different receptors. A receptor over here, and then this other one should go here. These are the receptors. It can bind to the beta adrenergic receptor, and it can bind to the alpha adrenergic receptor. And like I told you in the previous slide, if it binds to the beta adrenergic receptor, it will activate this pathway, the adenylyl cyclase pathway, right? Just told you, if it binds to the beta, it'll activate this pathway, adenylyl cyclase, right? All this stuff with, G protein, yes, alpha subunit activates adenylyl cyclase, and that's what gives you CAMP, right? And then the CAMP, remember, gives you now active protein kinase, correct? And let's pause right there and stop talking about the beta. We'll pause, and now let's talk about the alpha receptor. Alpha receptor is here, right? Epinephrine will bind to the alpha receptor, and ultimately it leads to the release of calcium, right? And that alpha receptor, how does it work? By the phospholipase C pathway, right? So just like I showed you up there, it's the first picture that I drew with the phospholipase C. It's by, how do you get all this calcium? This calcium comes from the release of the SR or ER, thanks to IP3. Remember, phospholipase C gives you IP3. IP3 binds to IP3 receptor, then calcium comes out of the ER. That's this calcium from the ER. From the ER. Okay, that's that picture up there. They're just doing a lot of shortcuts in this picture, which is that's the part I don't like, but that's why I covered it up there for you. And then what's going to happen to this calcium? It's up there again. That calcium is going to bind to calmodulin, right? Here's calmodulin. Originally, it's inactive, but if you bind calcium to it, remember I told you, it becomes active, right? And then calcium calmodulin activates protein kinase, and now you have, again, also an active protein kinase. So they both converge, they have that common product, which is active protein kinase. That's where they meet up. Everything after the active protein kinase is the common pathway. Okay? And eventually, from this from this active protein kinase, you break down glycogen and you end up with blood glucose. But there's a lot of little steps in here that we need to cover as well. And that's why we'll come on the board. So it's time to go back to the board. Okay? So, you're with me how we ended up with protein kinase, the active form, right? This came from the phospholipase C pathway, right? With all this calcium calmodulin, that's all of this. This is all the phospholipase C pathway. Anything involving calmodulin, right? And remember, this is this part. The calcium calmodulin complex gives you protein kinase, the active one, true. And then if you want, we can put the other pathway up here from the adenylyl cyclase pathway. And that one was, what activated this? Remember, it was the production of CAMP. We can even go further. This came from ATP, didn't it? What was the enzyme? Was it adenylyl cyclase? I'll just put AC. What bound to it to activate this enzyme? Alpha subunit. Okay. So now that's this is the adenylyl cyclase pathway. This is the phospholipase C pathway. And they converge at the active protein kinase, right? What do kinases do? Add what? Add a phosphate, right? So active protein kinase will phosphorylate, and it's right here. You're adding the phosphate to this enzyme called phosphorylase kinase. And this enzyme, when you phosphorylate it, it is active now. So obviously the previous one, the dephosphorylated or the unphosphorylated one, had to be inactive. So in other words, if you add a phosphate group to this, like here, you activate it. And now this is active, okay? Again, what do kinases do? Add phosphate groups, right? So this phosphorylase kinase will add a phosphate group to, it's right here, to this enzyme called glycogen phosphorylase. Before it was inactive when there's no phosphate. You add a phosphate to it, now you activated that enzyme, okay? Glycogen phosphorylase, the active form, takes glycogen and converts it into glucose 1-phosphate. It just, you get a glucose, but it still leaves a phosphate on it at the one position. The one position doesn't matter. But you see how I'm here, right? And then glucose 1-phosphate gets converted to glucose 6-phosphate, and then glucose 6-phosphate finally gives me my free glucose at the end. And each one of these has its own enzyme as well. You don't really have to worry so much about these two enzymes. This, for sure, yes. Okay, don't worry so much about this. It happens to be phosphoglucose mutase, and this one is glucose 6-phosphatase that gives you this. Okay, but you're with me how we ended up breaking down finally glycogen into glucose, right? So that's the whole goal. This is glycogenolysis, to break it down, not have any more of it, right? By breaking it down. Another way of not having any more glycogen is stop making glycogen in the first place, right? Over here, look at this enzyme over here, first of all. Look at glycogen synthase. The active form, which does not have a phosphate on it, it's a dephosphorylated enzyme. Glycogen synthase is what gives you glycogen. It takes glucose and it gives you glycogen. This is how you make glycogen, with this enzyme, the active form. If you want to stop making glycogen, how do you inactivate this? By adding the phosphate group to it. So when this enzyme is phosphorylated, it is inactive. Dephosphorylated or unphosphorylated, it is active, right? So doesn't it make sense that protein kinase is kind of doing two things? We know it's a kinase, that it's going to phosphorylate things, right? It's going to phosphorylate two enzymes. This one, phosphorylase kinase, phosphorylate it, so you can break down glycogen, and it's also gonna phosphorylate this enzyme right here. This is the second one that it does. Phosphorylates this enzyme so you can inactivate glycogen synthase, because if you do not inactivate it and keep it in the active form, you're gonna keep making glycogen. This is not what you wanna do. You're trying to break it down. You're not trying to make it. You need to stop this by adding a phosphate. Who adds the phosphate? Protein kinase. That's the first one from the common pathway.
So the active form of glycogen synthase doesn't have a phosphate?
Correct. If you phosphorylate it, the active form does not. If you phosphorylate it, then you inactivate it, which is what you want for glycogenolysis. You don't want to make it anymore. Because if you keep this active, you end up making the thing that you're trying to break down. So you need to stop making this. And the way to stop making it is add a phosphate to it. You will inactivate it if you add a phosphate. And that's what protein kinase is promoting, glycogenolysis. So you can promote glycogenolysis by obviously doing glycogenolysis, but also by stop making glycogen. So this enzyme right here does it phosphorylates these two enzymes, this one and that one. But the black one here, this one is for breaking down glycogen, right? So what this is also telling you is that you cannot make the generalization and say all phosphorylated enzymes are active. Not true. How about this one? This is phosphorylated, look, it's inactive. But yeah, most of these other ones, they happen to be active. Phosphorylated active. But yeah, this one is inactive. So phosphorylation just means there's a phosphate on it, that's it. It has nothing to do whether they're active or inactive enzymes. It just depends on the enzyme. This one, when it's phosphorylated, it's gonna be inactive. This one, these other ones, they're active when they're phosphorylated. Okay, and that's how glycogenolysis works. By epinephrine binding to two different receptors, two different pathways. The phospholipase C pathway and the the adenylyl cyclase pathway. It's not that bad if you study it, look at the picture and stuff, yeah. Adenylyl cyclase.
Is that beta or alpha?
This is alpha.
Oh.
This is like the beta? No, this is alpha, sorry. No, beta is this. This is alpha.
I think I'm confused.
The Oh, no, I see what you're doing. This is the alpha subunit. Okay, I was like, whoa, I'm like, wait a minute, I'm like, oh my God, you're confusing me. I meant the alpha subunit from G protein, not the alpha receptor. Those are different. The alpha receptor has nothing to do with the alpha subunit from G protein.
So those protein kinase does both of those at the same time?
Pretty much, yeah. It has to ensure that you don't undo what it already did by because here it's breaking down glycogen, and then here it has to make sure that it doesn't reform it, otherwise it's kind of like wasting your time. You're breaking it down, but then you're making it all over again. So you need to stop making this. Yeah? Any other questions? No? Okay. So let's move on here. So this is what I told you, right? The adenylyl cyclase is the one that gives you cyclic AMP, and the phospholipase C gives you the calmodulin. Again, I left my electric pen up here. Sorry. See, I told you I wore tennis shoes for a reason. I should stick my way back, right? Salvatory conduction. And this is all happening in the liver cell. Okay, and they converge, like I told you, at protein kinase, just like I had it up there. Okay. And again, those are my two receptors. But yeah, that alpha that I had up there was the alpha subunit. It wasn't the alpha receptor. Okay. Okay. So what I drew up there and kind of what this picture is saying is basically everything here in words. Okay, so I'm a picture person, so to me, I mean, all of this is accurate, so if you read through it, it does match the picture, but again, when you get something like this, you have to do something with it. You have to start doing, I'm fond of the curved arrow, because that means, it tells me one thing leads to another, and so forth, things like that, okay? But it says the same thing, right? Can you list all the enzymes involved in the converge pathway? They're right there, so yes, you can, okay? So I'm not gonna write them up, they're kind of right there for you, right? Can you draw the converge pathway? It's right there, I drew it for you, okay? You can redo it, I recommend redoing it, it's not the best way you can study this, right? Are the phosphorylated enzymes always active? No. We just said that it just depends. Some are and some are not. It just depends on which enzyme, you know. Okay? Well, the good news is we finished water-soluble hormones. Now we have to talk about lipid-soluble hormones next. They work a little differently, okay? The lipid-soluble hormones, obviously they're called non-polar hormones, right? Or they can also be called hydrophobic, sometimes they're called lipophilic. Sometimes lipophilic. Do I have it here? No, I don't think so, but yeah, lipophilic. And you know they can pass straight through the cell membrane. So receptor is not outside. You're not gonna see any of this cascade stuff. This cascade stuff that we did, that only happens for water-soluble hormone when the hormone stays outside. This is delivering the whole message inside now, because it can't go in, okay? And lipid-soluble hormones can go in, so their receptor is located inside, right? Either in the cytoplasm or in the nucleus. How do they work? A little differently. When the lipid-soluble hormones are traveling in the blood, they're actually bound to this carrier protein that literally carries them to the target cell. It's like a private taxi, and they take a ride on on the carrier protein, and they kind of take them right to the edge of the target cell, all right? And that hormone can go through, right? Because it's lipid soluble. Carrier protein does not go through though, right? So then the hormone enters the cell because it's lipid soluble, but I told you, not the carrier. The carrier actually will go back and pick up another hormone to deliver to the target cell, all right? If you were curious, it's not that important. The carrier protein is actually protecting the lipid soluble hormone, because you have to understand the lipid soluble hormone is in the bloodstream where it's like a liquid, watery environment. Do these like water? No, so it has to be kind of shielded and protected from the aqueous environment of the blood. So that's why it's kind of encased in a carrier protein. Once inside, that hormone is looking for its intracellular receptor that's on the inside, right? And again, it can be in one of these two places. And then once it finds its receptor, again, we're gonna have like a series of intracellular events taking place that ultimately is gonna affect gene activity at the DNA level, right? And here's like big picture, of what's happening here, right? So we know that the hormone is gonna end, so here's the carrier protein, like I was telling you about. Here's the lipid soluble hormone. Protected and carried, transported here, or carried here to the edge. And now the hormone can enter. And it's inside here now, right? Carrier protein can go back and pick up another one. All right? So this is what the whole goal is when you're dealing with lipid soluble hormones. The hormone receptor complex, the hormone is going to bind to a receptor. So this hormone receptor complex somehow must be present in the nucleus. It has to make, they both together have to make their way to the nucleus. That's the goal here. All right? So keep that in mind. So far, the hormone just got in and that's it. Here's the receptor for that hormone. This whole thing, don't worry about the light green and the dark green. The whole thing is the receptor. Okay? It found its receptor. This receptor is in the cytoplasm, obviously. It's not in the nucleus. I'll do another slide where the receptor this time is in the nucleus. But for now, let's say the receptor is in the cytoplasm. So hormone will bind to its receptor, and now we have this hormone receptor complex. Hormone receptor complex. Hormone found its receptor, but they're still in the cytoplasm. We're not done here. Didn't I tell you what the goal is? You have to be in the nucleus. So if the receptor and the hormone are still outside, bound together, they have to translocate into the nucleus. Translocation just means moving into the nucleus. Okay, so together they will travel through the nuclear membrane into the nucleus, and now the hormone receptor complex will bind to DNA. It will bind to DNA. Can't get that thing to turn on. It will bind to DNA, and then all this change in genetic and gene activity will happen. Okay, so like I said, it's right here, and this purple is my DNA. But now the hormone receptor complex found its way into the nucleus, right? Okay, and that's big picture. But again, we need to talk about the details about how this is actually happening here at the level of the nucleus. Okay, which is the next slide. All right. So this part I already just explained to you. I already kind of told you this part again, that lipid soluble hormone finds its receptor and you form this hormone receptor complex, right? And I told you have to make your way into the nucleus, right? And then together they get translocated again into the nucleus, right? Nothing new there. Now you're in the nucleus. Okay, so let me kind of explain to you big picture again what is happening here, right? Let's look at a piece of DNA. This long thing, pretend this is DNA. DNA. And then if you recall certain sections of DNA, pieces of DNA, this is what we call genes. So this is a gene, I'll just call it gene number 1, whatever, gene number 2. Gene number 2. And let me change colors. The area right before a gene, this little area, these are called promoters. So this is like a promoter region. This is a promoter region, and they're needed basically for gene activation, this promoter region. That's what their purpose is, right? Within the promoter, you have something that's called a hormone response element. There's a hormone response element made up of 2 half sites. Half sites made up of I don't know what I wrote here. Oh my goodness. Okay, this is made up of 2. I think that's what I meant. Of 2 half sites. Made up of 2 half sites, right? So it's basically, if this is this whole big thing here. Let's say this is my hormone response element, HRE. Am I in your way? My HRE. Then each little piece here, half of each. This is 1 half site here, and here's the other half site. Okay, and all of this is within the promoter region. Okay, so that's what again, like I told you, the HRE is found within the promoter. I told you that already. And again, 2 half sites make up 1 full hormone response element, and then you have in each half site, this is where you have the hormone receptors bind to it. Let's do green. So each half site, the flag looking thing is the receptor. Let's say this is the hormone. This is the receptor. At each one you have a hormone receptor complex binding to each half site. So you need 2 of them. Okay, I'll show you a nicer picture on the next slide. When you have 2 of them together, they kind of bind to each other. They're linked to each other. And and you call this now a dimer. Dimer means two, right? Because it's two, why have them here? It's two hormone receptor complexes. If there happen to be the same one, you call this homodimer. Later on, we're going to talk about a heterodimer when they're different. But if they're the same, the prefix homo means the same. So this is a homodimer. Over here, like the one I drew for you, right? Homodimer. And these homodimers are the ones that are responsible for activating the gene that leads to transcription and translation. Okay, so you see they work very differently from water-soluble hormones. Okay, the next slide has a nicer picture than the one that I just drew for you. Okay, ready? So again, I have my, they say it's steroid hormone, it's the same thing, they mean lipid hormone. You have your lipid hormone binding to its receptor, this whole big giant thing, the dark green and the blue and the light green, who cares about these two parts? Don't worry about this. The lipid hormone binds to its receptor, right? So, you know, I have now my hormone receptor complex, right? And it's gonna bind, remember, to a half site. And I have another one that's gonna bind to this half site, right? Now they're both here, right? And remember, both the half sites make up the HRE, hormone response element. And now they're both here, they're identical, so yes, dimerization, this is not just a dimer, but a homodimer. That form, because they're identical, right? Again, my two half sites, these two light purple ones are my half sites. Okay? And that, you know, stimulates this whole process of transcription and translation. When you have the two dimers occupying the entire HRE. Okay, occupying both of my half sites. Okay, any questions so far? You're good? Okay, you're good? Okay, alright. So that's how lipid soluble hormones, so you see they work a little different. There's none of this pathway. This whole pathway stuff, that's for water soluble. All of this, you know, HREs and the half site, the promoter, that's lipid soluble stuff. Easy to distinguish between the two. Alright? Let's talk about thyroid hormone a little bit. We haven't talked enough about thyroid hormone. Thyroid hormone is just by looking at this picture, you know it's not water soluble. There's no cascade of pathways, so you know it's not lipid soluble, so it must be, you know it's not water soluble, it is lipid soluble. Okay, so this is a lipid soluble hormone, right? So same thing, it's lipid soluble, so it must require a carrier protein to take it to the cell surface, right? So, and that happens to be T4 though, that the carrier protein binds or carries and binds to, right? And then T4 finally enters the cytoplasm. And then remember we said T4 is inactive, but T3 is active, so inside the cytoplasm, this is where T4 gets converted to T3, inside here. You see it right here, right? T4 gets converted to T3. Who cares about this binding protein? Don't worry about this green binding protein. The receptor for T3 is automatically always found in the nucleus though. So you see the receptor, the orange receptor, it's a receptor. So its receptor is not a cytoplasmic receptor. It's already in the nucleus. So that's why I'm talking about this picture. Remember the previous picture, this picture talked about if the receptor is in the cytoplasm, that they have to translocate. Now with this other similar picture, the receptor is not in the cytoplasm, but it's now in the nucleus. That's why we're talking about this, to give you a perspective of how things happen when the receptor is in the cytoplasm versus in the nucleus. Here, the receptor is in the nucleus for thyroid hormone. Okay, thyroid hormone T3 makes its way inside the nucleus, looking for its receptor, which is already there. Okay, and the same thing's going to happen. The hormone receptor complex, you know, the T3 and the T3 receptor are gonna bind to a half site of the HRE, which is what's happening here, right? This is the TR, this is for TR3. I'm sorry, for T3. Triautothioamine is TR3 receptor. Right, I keep saying TR3, T3 receptor. Right, it's this orange one. And then thyroid hormone comes in, binds to it, right? And occupies one half site. The light purple is one half site. What about the other half site? You don't require another thyroid hormone to activate this DNA. All you need is one. But you need a partner. It needs a partner. The partner that it's going to get has nothing to do with thyroid hormone. It's a receptor called the CXCR receptor, and the thing that binds to that receptor is this other weird thing called 9-cis-retinoic acid that binds to it instead. And now you still form a dimer, because both of these are going to link to each other, but are they the same one? So it's not a homodimer, it is a heterodimer. Heterodimer. Because they're different. Okay, so the counterpart is the RXR receptor with 9-cis-retinoic acid. Okay, and then otherwise then it's the same thing as the other one. So these were two examples when you had a receptor that was in the cytosol versus in the nucleus, right? The thyroid hormone receptor is found always in the nucleus, it's not in the cytoplasm. You ready for the next one? That was it. Okay, I forgot to put my good life slide, I always do that, but I haven't had time to, I'm gonna catch up with what I need to do. Okay, so that was the end of that. Okay, finish a little early, any questions on any of the material? We still have our session on Friday, and I plan to go over all of the hormone stuff, probably do some stuff like this in here. Okay, so I will have some questions out for sure tonight, I won't finish probably the entire chapter.